Yes, everyone. So I hope that I am live and I am clearly audible and visible to you. Just let me know in the comment section if you are able to hear me, if you are able to see me. Yes. Yes, guys. Yes. Hi, Shivesh. How are you? Good to see you. Very good to see you, Shivesh. Okay, great. I am audible and I am completely visible to each and every one of you. So today what we are going to do is that we are going to start with the fifth lesson, fifth class of this particular chapter. And in today's lesson we are going to try to complete the transcription. Okay, Shivesh? Anyone in the comment section, let me know. Great. So chalo, let's quickly start the session. So what things we have discussed in the last class? We have discussed few of the things which was translation. DNA replication. We discussed one topic which was DNA replication. Yes or no? I hope that you remembered, guys. I know the class is being after a long day. We are having a live class, but I hope so that you all remember this. So what we are going to discuss today. Before that, I am going to make you revise the things. So we discussed about DNA replication. Yes or no? We discussed about DNA replication. So this DNA replication was what? This DNA replication, we discussed that it was semi-conservative. Yes or no? It was semi-conservative, right? And what? And we also discussed the whole process. We discussed that there is some DNA A protein which is involved. Yeah, right. DNA A protein which was involved, and this protein was responsible for what? This protein was responsible for the separation of the two strands of DNA. Yes or no? And after that, what happened? After that, when this protein was transferred, then there is an enzyme called which was involved, which is DNA dependent DNA polymerase. Yes or no? DNA dependent DNA polymerase, right? DNA dependent DNA polymerase. This enzyme was there. Okay. Then we saw that there was two types of strand formation, right? Let's suppose that this was a DNA. Let us suppose that this was the DNA which has to be replicated. So there were two strands. One was what? One was synthesized regularly without any lag. So this strand was known as leading strand, and the other strand was what? It was synthesized in opposite direction, so it was known as lagging strand. Yes or no? It was known as lagging strand. Clear? Next what, guys? Next we also discussed that these were the Okazaki fragments. So इतना कुछ हमने last class में discuss कर लिया है. So today what we are going to do is that today we are going to start with the experiment which proved that DNA replicates by the semi-conservative. Okay, we are going to have an experimental proof for what? For experimental proof for semi-conservative nature of DNA. Semi-conservative nature of DNA. Okay, so let's see कौन सा experimental proof है जो ये DNA को बताता है, like proof करता है कि yes, DNA replicates through the semi-conservative nature. Yes or no? Okay, great. So see here, there were two scientists which was involved, and the scientist name was what? Stiles and Melson. Okay, there were two scientists which was what? Any doubts in the comment section, guys? Let me know. Okay, if you have any doubts, let me know in the comment section. So we have two things, which is Melson and what? And Stahl. Okay, these were the two scientists who proved that DNA replicates through the semi-conservative nature. Now how it proved? See, so they took what? They took E. coli. The experimental organ for them was E. coli. Okay, so they took E. coli here. Now what they do is that they allowed this E. coli. This is the normal E. coli. Okay, now they allowed this normal E. coli to grow into N14 medium. Okay, to grow into N14 medium, right? Now many of you guys have this confusion that, sir, whether this N14 medium is radioactive or not. So guys, this N15 medium is non-radioactive. it's non-radioactive. Okay, so what they did, they just took the E. coli, and this E. coli was allowed to grow in a china dish which contained N15 nitrogen. Okay, which contained N15 nitrogen, right? Okay, great, sir. Now when this E. coli decided to divide its DNA, decided to replicate its DNA, so how it happened? See, so this was the normal E. coli. Okay, I'm going to make here, this was the normal DNA of E. coli. Okay, normal DNA. Now then what? Then this was allowed to grow into N15 medium. Yes or no? This was allowed to grow into the N15 medium. Now what happened? See, when it was allowed to grow into N15 medium, and this E. coli started to divide. Hi Bala, how are you? This E. coli started to divide. Then what happened? See, when it formed the another strand, here, so what happened? We found that the another strand was containing the N15 nitrogen. The another strand was containing the N15 nitrogen. Yes or no? Yes or no? See here. Is that so, Bala? Okay, 1 second. I'll make it short. 1 second. 1 second. Okay. Bala, adjust it today little bit. Okay. आज थोड़ा सा adjust कर लो. After today, we will be back with the same setup. Okay. After today, we will be back with the same setup. Okay. Okay, Bala. चलो. Great. Should I start? Okay. Am I lagging? Okay, chalo. So now what happened? Now what happened, guys? See, we had this E. coli. So this was the normal E. coli. Yes or no? This was the normal E. coli. This normal E. coli was allowed to grow into the medium which was containing the N15 medium, right? N15 medium. Okay. Now after 20 minutes, we all know that E. coli divides in 20 minutes. Yes or no? E. coli divides in 20 minutes. So after 20 minutes, what happened? We saw that DNA, that these people saw the DNA, and they saw that the another strand, the newly synthesized strand, was containing N15 DNA. In the similar manner, they again gave the time. They again gave some time, like again after 20 minutes, they again checked what's happening, guys. Yeah. After 20 minutes, they again checked. So see, this was the DNA. So what happened? They got like this, they got separated, and again the new strand was formed. So it was having again what? It was having N15 medium, again N15 nitrogen. So this was the proof which was saying that DNA is what? DNA is replicating through semi-conservative nature. Yes or no? Any doubts, guys, anywhere? Any doubts, guys, anywhere? Let me know in the comment section. Any doubts anywhere? Okay, now how they separate? See, they want to what? Any doubts, guys? Should I move forward? Okay, great. So we left this part in the DNA replication thing, and now let's start the transcription thing. Okay, now let us start the transcription thing. I hope that there is no doubt. Okay, chalo, great. Let's start this session. Let's start it. Okay. Now before starting the transcription, I would like to introduce to the RNA. What is RNA? RNA is ribonucleic acid. We all know, sir. We all know, right? And if I talk about the structure, so it is same as DNA. It is same as DNA, but there is one thing which is different here. What is that thing, guys? Bala, what is that thing? What thing makes RNA and DNA different? Number one thing that in RNA, in RNA there is presence of ribose sugar, yes or no? Ribose sugar, presence of ribose sugar, while in DNA there is presence of deoxyribose sugar, yes or no? Yes. After this what? After this there is one more difference, which is the presence of uracil, right? So in RNA uracil is present, and in DNA what is present? In DNA thymine is present. So these are the two major difference between the structure of RNA and DNA. Any doubts anywhere? Any doubts anywhere? Yes or no? Okay, chalo. Now in most of the cases, this DNA acts as a genetic material. This DNA acts as a genetic material. Yes or no? We all know this. DNA acts as a genetic material. Yes. But guys, there are few exceptions. You know the exceptions? There are very few exceptions, which is what? Which is in reovirus. If I talk about the reovirus, okay? If I talk about the TMV virus, which is tobacco mosaic virus, okay? And if I talk about the bacteriophage, so what is the genetic material, guys? इसमें genetic material क्या है? What is the genetic material? This is the RNA. The genetic material in all of these things are what? This is RNA. Yes, yes, very good, Shavesh. This is the RNA. Clear to everyone? Is it clear? Okay. Now, so basically the RNA is of two types. Again, RNA is of two types. One type is making the genetic components of the body, is acting like a genetic component, and the second component is what? Second component is acting like, one second, what happened? 1 second, bachcho, 1 second. Something happened to my slide. Just a second. Why this is not being removed? Sometimes this slide is like show tantrums. 1 second, bachcho, 1 second. What's happening? Just a second, guys. ओहो, क्या हो गया? Just a second, बच्चों. This part is not being removed. Just a second. I'll try once again. I'll try once again, bachcho. 1 second. Okay, something happened. I will restart. Okay, I'll refresh it. Just a second. ओहो, क्या हो गया ये? Okay, 1 second, guys. 1 second. Just give me a second, okay? Just give me a second. I will restart this. 1 second. There is some issues. Till then, let's revise something. एक बार मैं इसको वापस से स्टार्ट कर दूं, फिर इट विल बी फाइन. ओके, देन इट विल बी फाइन. ये क्या हो गया बच्चों? Just a second. I'll be back. Yes, Shivesh, I will be just 1 minute. Just a minute, okay? Just a minute. Okay, so guys, till then, see what we were discussing about the RNA, right? We were discussing about the RNA, and RNA was what? And RNA was something which can act as a genetic material. Also, we saw that in some cases, the RNA is acting like a genetic material. Yes or no? Yes. But in some places, now it's clear, na? I think so. It's clear. Yes, it's clear now. Chalo. The issue is resolved. I don't know what happened, but ठीक है. Chalo. Great. So yes, what we were discussing that RNA is what? RNA is different to DNA, and we discussed the differences like RNA contains uracil in place of thymine, yes or no? RNA contains what? RNA contains the deoxyribose sugar, yes or no? So these things, sorry, RNA contains the ribose sugar, RNA contains the ribose sugar. So these things we have discussed. Right? Now what happened? Like we have seen that RNA is what? RNA in some cases it is acting like a genetic material. In some cases it is acting like a genetic material. So what we say is that we say RNA as genetic RNA. So we have given two types of RNA. We have given two categories of RNA. If the RNA is acting like a genetic material, we call that RNA is genetic RNA, okay? And there are some RNA, or I should say there are some major RNA which is not acting like a genetic material. So we call them as non-genetic RNA. We call them as non-genetic RNA, okay? Non-genetic RNA. Now, if I call this non-genetic RNA, guys, so these are of again three types. rRNA, okay? And after this we have tRNA, and then we have mRNA, right? Okay, so what things we discussed? We discussed that RNA is there. RNA can act like a genetic material, and if it is acting like a genetic material, we call it as a genetic RNA. And then we have a non-genetic RNA, and the non-genetic RNA are what? They are the rRNA, tRNA and mRNA. Now, I am going to give you a very basic functions of all of them, okay? बहुत ही basic सा function में तुम्हें समझाने की कोशिश करता हूँ. See, what happened? We are talking about the transcription, right? We are talking about the transcription. So what happens in transcription? See, we have made a DNA, right? We have replicated a DNA, right? So this DNA is going to make what? This DNA is going to make RNA, yes or no? So the process of formation of DNA, the process of formation of RNA from what? From DNA is only the transcription, right? Okay, clear? Yes or no? Any doubts anywhere? Let me know in the comment section. Any doubts anywhere? Let me know in the comment section, bachcho. Okay. So this is what? This is the basically the transcription. So in transcription, we are going to make what? We are going to make the RNAs. Okay. Now let's see how we are going to make the RNA, and after that, after we have seen how they are made, we are going to talk about their properties. Clear? We are going to talk about their properties. Ready, everyone? Ready, everyone? So we saw DNA replication, right? We saw DNA replication, and DNA replication process was what? It was same in prokaryotes and eukaryotes. DNA replication was same in prokaryotes and eukaryotes. Okay. Okay. So that's why we have studied it all together. Okay. Same in prokaryotes and eukaryotes. But guys, you know, if I talk about the transcription, it is what? It is different. Why it is different? I'm going to tell you. First of all, let us start with the prokaryotic transcription. Okay. First of all, let us start with the prokaryotic transcription. Prokaryotic transcription, or you can also say it as bacterial transcription. Okay. Bacterial transcription or prokaryotic transcription. See, remember guys, remember that prokaryotic transcription is different from eukaryotic transcription. Transcription is both different from for eukaryote and prokaryotes. Unlike DNA replication, it was same. The process was same. Whether it's prokaryote or eukaryote, it was same. But transcription is different for both prokaryotes and eukaryotes. Clear to everyone? Yes or no? Yes or no? Okay. Bala, are you there? Bala, if you are there, reply me. Okay. Any doubts? No doubts? Okay. Great. Great. Chalo. Chalo. Now, let's move on. Let's move on. If I talk about the prokaryotic transcription, so how the prokaryotic transcription is occurring. See, so we have a gene. We have a DNA. Okay. Let's suppose this is the chromosome. Okay. Let us suppose that this is the chromosome. Right? This is what? This is the chromosome. And If I talk about the chromosome, inside this chromosome, what do we have? We have the DNA. Yes or no? DNA genes are there. So I'm going to take out the DNA. Okay, I'm going to take out the DNA. So DNA will be what? DNA will be like this. DNA will be like this. ये कैसा बन गया? हाँ? ये कैसा बन गया? चलो. DNA will be what? DNA will be like this. Right? Okay. So now what? We have to do the transcription. Now we have to do the transcription. So to do the transcription, let us make a easy structure of DNA. So suppose that this is the DNA. Okay. This is the 2 strands of DNA. Here we are talking about what kind of DNA? Here we are talking about the double strand DNA. Okay. dsDNA. Here we are talking about the dsDNA or double stranded DNA. Clear? Now what? Now let's suppose that this DNA is from 3' to 5'. This segment is basically from 3' to 5' and this segment is from 5' to 3'. This segment is from 5' to 3'. Right? Now see, this whole structure is known as what? This whole structure is known as the DNA. Right? ये दोनों क्या है भाई? DNA है. Yes or no? This is DNA basically. Now see, very carefully guys, very carefully. From this DNA, we are going to make what? We are going to make RNA. इस DNA से हम क्या बनाएंगे? हम RNA बनाएंगे. Okay. So the enzymes which we are going to use, will be what? Will be of this particular process. You remember in DNA formation, we were using DNA dependent DNA polymerase. You remember? DNA dependent DNA polymerase, right? DNA dependent DNA polymerase we were using. But in this case, what we are going to do? In this case, we are going to use some other enzyme which is DNA dependent RNA polymerase. DNA dependent RNA polymerase. Why we are going to use this enzyme, sir? Why we are going to use this enzyme? Why we are not going to use this enzyme which is DNA dependent DNA polymerase? See here. So in first case, when we were replicating the DNA, what we were doing? We were copying the DNA. And making the DNA, right? So here we were forming the DNA with the help of DNA. But here what we are going to do is that we are forming the RNA with the help of DNA. Yes or no? So that's why the enzyme which we are going to use is DNA dependent RNA polymerase. Okay, the enzyme which we are going to use is that DNA dependent RNA polymerase. Clear to everyone? Yes or no, bachcho? Let me know in the comment section if you have any doubts. Let me know in the comment section if you have any doubts, guys. No doubts? Great. So now you are clear why we are using this DNA dependent RNA polymerase. Right? So first enzyme which we are going to use here will be DNA dependent RNA polymerase. Okay, 1 second. I will be adding some slides. Just give me a second. I'll add some slides. Okay. I will add some slides. Okay, bachcho. Chalo. Now it's done. So till now we have seen DNA dependent RNA polymerase, right? अब देखो क्या क्या चीजें चाहिए हमें. Now what things we need? Second thing is what? This DNA dependent RNA polymerase is a very simple enzyme. DNA dependent RNA polymerase is a very simple enzyme. How, sir? See, you remember that DNA dependent DNA polymerase said that I need something, I need promoter, I need the sequences, I need the primase, I need the first sequence, I will just work in this direction only, this, that. So there were lots and lots of demand of DNA dependent DNA polymerase, but this DNA dependent RNA polymerase has no demands. It's a very simple enzyme. It is going to do everything. सारा काम DNA dependent RNA polymerase कर लेगा. बस इसको कुछ चीजें चाहिए. A very bare minimum requirement. See, so suppose that it is going to make the RNA from this particular segment of DNA. Okay. So it says what? It says give me a promoter box. Okay. Give me a promoter sequence. To enter into the transcription, I need something which is what? Which is the promoter. Okay, which is the promoter. Clear? So here what we need? A promoter box. Okay, this thing requires a promoter box. Okay, great sir. Now this DNA dependent RNA polymerase doesn't know how to start and doesn't know how to end. So to start also it needs help with the promoter, and to end also it needs help with the terminator. Okay, with the terminator. So two things we are required now. What sir? One is the promoter and the other one is terminator, right? Okay, great sir. Now so what about the polarity here? So see here, first of all, this DNA is what? This DNA is 3 prime. This segment of DNA is from 3 prime to 5 prime, and the other strand will be what? It will be 5 prime to 3 prime. Yes or no? Yes sir. Yes. Any doubts here? Any doubts here? No doubts. Great, great. No doubts. Okay, great. Shivesh, any doubts? Shivesh, Bala, any doubts? Okay, great. No doubts. Now see, I'm going to give you a very good thing. You won't forget anything after this. Okay, so see here. Now the scene is that you will see the promoter. Okay, you will see the promoter first, and then you will see the polarity. So see here, if the promoter is what? If the promoter is being attached from 3 prime, if this segment on this segment, you see first what you see? First you see the promoter. Okay, पहले किसको देखना? पहले promoter को देखा हमने, ठीक है? Promoter को देख लिया. Yes, this is the promoter cell. Now you check the polarity of the strand. Now you check the polarity of the strand. Okay, so see here, this is what? This is 3 prime to 5 prime. So the DNA segment, the DNA strand which is going to have the polarity from 3 prime to 5 prime, that DNA segment is called as what? It is called as template strand. What we are going to say? Template strand. How did we recognize that? We saw the promoter, position of promoter we saw. Okay, great. We saw the position of promoter, and after that we check the polarity of the strand. After that we check the polarity of the strand, and if the polarity of the strand is 3 prime to 5 prime, then that strand will be called as the template strand. Okay everyone, yes or no? Yes or no? Any doubts? Any doubts guys? Any doubts? Let me know in the comment section. Okay, okay, great, great, great. So this is how we are going to recognize the template strand. So remember, template strand is 3 prime to 5 prime on the basis of the position of promoter. If suppose I am going to, one second, something happened. I don't know why this issue is coming again and again, but I'll try to figure it out. Just a second guys. There is an issue. One second bachcho, one second. I'll again restart. Okay, I will again restart it. There is some issues going on. Actually, I'm not able to remove this thing from every slide. It is coming. Just give me a second, bachcho. Just give me a second, okay? Chalo. So, suppose that I am giving you, till then let's revise something. So, what things we have discussed? We have discussed that if the promoter is present, first of all, if we have to identify the template strand, what we are going to do? I am going to see the position of promoter. Now, the position of promoter I have seen. Now, check the polarity. If the polarity is from 3 prime to 5 prime, then that is the template strand. So, sir, if that is the template strand, what will be the opposite strand? How we are going to call, what we are going to call the opposite strand? Yes or no? So the opposite strand is called what? It is called as the non-template strand. It is not going to make any template. It is not going to make any RNA. So we are going to call it as the non-template strand. Clear to everyone? Yes or no? Yes or no? Okay, just a second. Now my PPT is again open. Just give me 1 second. Just a second, बच्चों. Okay. Guys, I am facing some technical issues. I don't know why I am facing. I'm not able to let's see, let me check. It's lagging guys, it's lagging. So am I able to write here? Let me check. What's happening? Okay, see, let's आज का session we will end here only. Okay, till now whatever we have discussed, I'll again schedule a class for transcription. Okay, I'll again schedule a class for transcription, and in that we will study from starting. I'll revise whatever the things I have made you studied in this session, and then we will complete the transcription. Okay, I think so there is some issues which is going on and I am not able to solve it. So instead of wasting your time, let's end this session. Let's meet tomorrow at 6:30. Okay, let's meet tomorrow at 6:30 everyone. Okay, whoever is watching this video, let's meet tomorrow at 6:30. Okay everyone, clear? Okay, I'm so sorry guys, I'm